Thank you, John. That was actually pretty much sums it up. I wanted to talk today about some work that I've done looking at the concept of lipotoxicity as sort of a general big picture model of why this matters or how it all goes together. First I should say I have no disclosures. And I just want to start by acknowledging some of the people who contributed to this work and were involved in my training as well as in the research that was done, particularly also Dave Bernlohr, whose lab I've been working in for the last several years, and Alan Senayko, who is the PI of a major study that I've been doing sub-studies on and has been also a mentor. And then, of course, the collaborators in the lab and elsewhere. So the objectives of this talk, first I want to talk about, so the idea is to start with small and work our way up to big, and then in the end you get the treat of the clinically relevant information. So the beginning is to look at specifically at the adipocyte and look at some basic mechanisms connecting adipose tissue oxidative stress with inflammation. And then we're going to move to the translational, looking at the role of free fatty acids in the development of insulin resistance. And then finally, I want to talk about the evaluation of insulin resistance and diabetes in obese children. So starting with our first, all right, so lipotoxicity. So here we have the adipocyte. Let's see if I can find there we go. The adipocyte with the triglyceride droplet, which makes up the vast majority of the adipocyte. And in the past, people thought of adipocytes as being sort of a passive storage depot for the extra energy you get during your food intake. But more and more over the last 15 years, people have begun to realize how important the adipocyte is in the whole body energy metabolism and the development of metabolic disease and cardiovascular disease particularly. So we know that with obesity, there's an increased production of reactive oxygen species, ROS, and that's a state called oxidative stress. And with that, there's evidence that you have oxidative stress is associated with an increased rate of lipolysis. So the adipocyte itself becomes insulin resistant to the anti-lipolytic effect of insulin, and that's actually at a lower level. The anti-lipolytic effect is at a lower level of insulin than the amount of insulin you need for uptake of sugar into muscles, for instance. So the concept of lipotoxicity, so people look at what is the connection between the adipocyte and the various metabolic effects. And there's lots of molecules that have been proposed as being critical, with adiponectin being one that gets a lot of press, and the other that many people have heard about is leptin, which kind of revolutionized the field of adipose biology, where people kind of recognize that, wow, there's something more going on here. But the free fatty acids are interesting in that you can connect free fatty acids mechanistically to a lot of the diseases that result or are associated with obesity. So, for instance, in the liver, free fatty acids are associated with increased hepatic glucose production and also with increased lipogenesis in the liver. And with increased lipogenesis, you have eventual development of non-alcoholic fatty liver disease, or NAFLD. With the beta cell, free fatty acids are associated with increased apoptosis of the beta cell and reduced insulin secretion. The muscle, this is the one that is most relevant to this talk. In the periphery, the muscles are less insulin sensitive in the presence of free fatty acids. And then finally, the endothelium is also affected by free fatty acids in that you have endothelial dysfunction and proatherogenic changes in the endothelium. So as I mentioned, we're going to kind of talk about that, the fat, muscle, insulin resistance axis. So important when you're talking about adipose tissue is to think about the fact that it's not just the adipocyte, that there are other really important cell types that are playing a role. So for instance, the endothelial cells in the microvessels, the tissue resident macrophages are also really important, and both endothelial cells and macrophages have been shown to play a role in both the regulation of energy storage and also the sort of systemic effect of obesity. So kind of getting down to the basic model. So you have the adipocyte. As you get obese, the triglyceride droplet gets bigger, and the adipocyte becomes stressed in a sense, and there's an increased production of reactive oxygen species, so increased oxidative stress. This is associated, as I mentioned, with increased lipolysis at basal. Also the production of adipokines and cytokines. So I'm just not going to focus on in this talk today so much, but that's also implicated in the connection between the adipocyte and the macrophage. So the macrophages, in response to obesity, are recruited. So you have more tissue-resident macrophages in insulin-resistant obese individuals in their fat tissue than you do in a lean. So the macrophages then produce inflammatory cytokines and mediators, and those then act on the the adipocyte to increase oxidative stress further. And then you have increased free fatty acids, et cetera. So it's kind of a vicious cycle. These free fatty acids then go to the periphery via the endothelium and have the effects of lipotoxicity I talked about previously. So looking at this process, I wanted to look most specifically at the adipocyte itself and how it plays a role in this pathophysiology. So the part of the Bernler lab has been interested in this process, and some of this has been delineated in model systems, and my interest was looking at how this relates to humans. So, but the first part is model systems. So we have polyunsaturated fatty acids that are part of the lipid membrane, and those get acted upon by reactive oxygen species. So there's a lot of effects of reactive oxygen species in the cell on DNA, on proteins directly, and so this is one of many deleterious effects of reactive oxygen species. So there's a series of steps whereby these polyunsaturated fatty acid chains from the lipid get transformed into what's called a reactive lipid, or a lipid peroxidation product, the most abundant of which is 4-H&E, 4-hydroxynonenal. And that is a lipid that has been activated. It's more chemically reactive, and it's able to covalently modify proteins at various residues, the histidine, cysteine, and lysine. So those are all able to be modified by these lipids, and then you have this little kind of lipid chain hanging off of the protein, and that can have a variety of effects. It's called carbonylation, and that can target the protein for degradation, but it can also have a protein that remains but has an altered function. And in the Bernler lab, this has been looked at in mouse and cell culture models where they've shown that carbonylation is associated with altered mitochondrial function, as well as altered basal lipolysis. So these fat cells with increased basal lipolysis are also have an increased level of protein carbonylation. And then, on the other hand, these reactive lipids can be metabolized. So there's an enzyme GSTA4 that conjugates these lipids to glutathione, and glutathione probably is familiar to people as an antioxidant. It has lots of roles, and this is one of them. So when you conjugate to glutathione, you get something called GSH&E, and that is then able to be transported out of the cell actively by a protein called RLIP76. And I just mention this protein because it's interesting. If you knock this protein out in mice, the mice can get fat, but they are resistant to, or they do not get insulin resistant. So they kind of get protected by just getting rid of this, this molecule. So based on that and other observations that have been done previously in other cell types, we hypothesized that perhaps this GSH&E isn't just a byproduct of metabolism, but actually has a functional role. And so the hypotheses were that, number one, we'd be able to show that GSH&E would be the product of oxidative stress in adipocytes. Number two, that GSH&E would have a functional role, that it would induce inflammation in the macrophages. And so this would provide a novel mechanism by which adipocytes are communicating with the macrophages, and you're connecting both the oxidative stress and inflammation in the tissue. And finally, that GSH&E would be increased in the adipose tissue of high-fat fed mice. So to look at this, first, I grew adipocytes in both low and high glucose conditions. And just to give people context, the low glucose is about a normal fasting glucose, and the high is quite high, 450. So the bottom is kind of what we think of clinically. And as you can see here from these are just two different magnifications. The fat cells here versus the fat cells in the high glucose fat cells have more and bigger lipid droplets, and that's been shown previously in this type of system. So then looking at these cells, we looked at how much GSH&E was made in these cells. And so on the left here, you have the control cells. And on the right are cells that are treated with hydrogen peroxide, which is induces oxidative stress. So in the control cells, between the low and high glucose, there was the high glucose, there's a little bit more GSH&E, but it wasn't significant. But if you look at the cells that have oxidative stress, the ones who are grown in the high glucose make considerably more of this product. So basically there's a synergy between high glucose conditions and oxidative stress in the production of GSH&E. So the next part of the project was, and we characterized this in other ways, looking at the genes that are important, et cetera. But moving on to what this means, the next part of the project was asking, does GSH&E actually have a function? And so what I did is I treated a macrophage cell line with varying amounts of GSH&E. So along the bottom here, you see the various concentrations, with this being the control. And along the y-axis is the measurement of how much TNF-alpha was produced by those macrophages in culture. And as you can see, there's a dose response, that the more of this GSH&E you put on, the more TNF-alpha you produce. And we also saw a similar effect with leukotriene C4, which is a inflammatory leukotriene. And then looking at the bigger picture in a whole mouse, so here we have low-fat diet mice. Here are high-fat diet mice, and this is the OB mouse model. So that's a leptin knockout mouse that gets very obese. And so you can see that in the obese mouse fat tissue, there's more GSH&E than in the lean mouse. So in vivo, we see that this relates to obesity. So that part of the project, we'd shown that adipocyte oxidative stress induces the production of GSH&E, and that GSH&E induces the production of inflammatory mediators by macrophages. So this is now the beginnings of a new signaling system between the two cell types in the fat tissue, and that GSH&E is increased in the obese mouse adipose tissue. And we've also looked at now some mouse models looking at their insulin sensitivity and have shown that there is some association with GSH&E, but that's still in preliminary stages. So the next part is looking a little more translationally, and this gets into the other side of the system where I talked about the carbonylation of proteins. So that's, instead of making the GSH&E, the reactive lipid goes on to modify proteins. And as I mentioned, that's associated with adipose dysfunction. So this has been previously looked at in model systems, and my question was, what happens in humans? So the hypotheses were that obesity would be positively correlated with amount of protein carbonylation in human subcutaneous fat, and that the carbonylation would be correlated with free fatty acid levels because of this connection with lipolysis that had been shown in model systems. And finally, that carbonylation would be inversely related to insulin sensitivity. So for this study, it was a pilot study. We had 8 individuals of average age 26, and they had a wide range of BMI. They had a frequently sampled IV glucose tolerance test to measure their insulin sensitivity, and they had a subcutaneous fat biopsy, which was done by Dr. Chowdhury, a plastic surgeon that has been our collaborator. So looking at the fat from these individuals, I measured protein carbonylation. So this is a sample blot. Let's see if I can get my... So here on the left, this is a lean individual. In the middle is the obese individual. And here is a negative control. And this is a little bit like a Western blot, if that says anything to anybody. But basically, each band there represents a protein that is carbonylated, so altered. And as you can see pretty clearly, in the lean individual, there's a lot less protein carbonylation than in the obese. And down here below is the protein loading control, showing that there's the same amount of total protein in each lane. And so when you looked at the 8 individuals and quantified the total amount of carbonylation, you see that as BMI goes up, the amount of carbonylation in the tissue goes up. And we found a really tight correlation there. So additionally, I looked at the relationship of carbonylation with free fatty acid levels in the serum, and again showed a really tight correlation between the two. And this appeared to be independent of adiposity, so it wasn't just a matter of those people being more obese. And then the last part was looking at, is there a relationship between carbonylation and insulin resistance? I'm not going to show you that data because we didn't see a relationship. It looked like with a bigger N, more than 8 people, we might be able to find something there. There was kind of a trend, but not significant. So the summary for this little part is obesity is positively correlated with protein carbonylation in the human subcutaneous fat. That carbonylation is correlated with free fatty acid levels. And finally, it remains to be seen whether carbonylation and insulin resistance correlate in a human population study. So, kind of recapping, we've gone from the adipocyte showing that this change in lipids from oxidative stress has both signaling potential as well as potential to alter protein function in the adipose tissue. And both of these have implications in terms of the whole organism and how that will affect insulin resistance, et cetera, either via free fatty acid levels or alterations in adipokines, so signaling molecules from the adipocyte. So that's a whole other area that could be looked at. So now kind of moving into the translational, the question was, looking at the role of fatty acids in the development of insulin resistance. So we know that if you take an individual in a research setting and you infuse them with intralipid and you measure their insulin sensitivity before and after with a clamp, you'll see that their insulin resistance will increase with just the presence of more lipid in the circulation. There's also the converse experiment that's been done where people who have high lipid levels, high free fatty acid levels, are treated with medications to drop the free fatty acid levels, and they see before and after an improvement in insulin sensitivity. So there is an association in experimental models. And then there's also some data in adults that people with a genetic history or propensity to diabetes have higher free fatty acid levels compared to people with the same BMI but without a family history. So our question was looking at Alan's longitudinal study where he has data from these people. So these are participants who were recruited at a mean age of 13 from Minneapolis public schools, and they've now been studied for almost 20 years. These individuals are in a longitudinal study of obesity and insulin sensitivity. So we have data from multiple visits, and particularly for this study, we're looking at visits at age 15 and age 22. And as represents the population locally, most of these individuals were white. So that's one limitation to this study. The parents of the subjects were also recruited during the course of their children's participation. And so we used the parents not so much because of their relationship to the subjects themselves, but as an older adult population, as a comparison population. And we looked at the insulin resistance measurement by euglycemic hyperinsulinemic clamp, which is denoted by M. And so we talk about M as a measurement of insulin resistance, but just intuitively, it's easier to think of it as a measurement of insulin sensitivity because as N goes up, the person is more insulin sensitive. So just to kind of keep that in mind. So higher M is more insulin sensitive. So our hypotheses were that the free fatty acid levels in these individuals would be associated with their insulin resistance and that this relationship would increase with age. And then secondly, because we have longitudinal data from 15 and age 22, we predicted that the free fatty acid level earlier would predict later insulin resistance into adulthood. And so just a little bit of information about the subjects. There was these first two columns here are the same individuals measured at age 15 and age 22, whereas this third column here is the parents, which is the kind of older adult comparison group. And so as you can see, of course, the age goes up. The gender of the parent group was more female. And all of them were predominantly white, but the parents being a higher percentage of white as compared to African American. And then BMI went up with age, which unfortunately is sort of what you see in population studies. As we get older, we go up in BMI. And then the insulin sensitivity was higher in the adolescents relative to when they were young adults and their parents. And then finally, the free fatty acid levels was higher in the adolescents versus later in life. So when we looked at the relationship of their free fatty acid levels with their insulin sensitivity, we saw that at age 15, it seemed like there was a general trend that as the free fatty acid levels were higher, people became less insulin sensitive, but that wasn't significant. On the other hand, if you looked at the same individuals at age 22, now there was a significant relationship between free fatty acid levels and decreasing insulin sensitivity. And in the parents, the relationship was even stronger. So with age, that relationship is increasing. All right, but next to look at, in the individuals that we can look at age 15, and again at age 22, Along the x-axis here, we have the free fatty acid levels at age 15. And on the y-axis, we look at their insulin sensitivity at age 22, and looking at whether you can predict the later insulin sensitivity from the earlier free fatty acid levels. And you see that there is a significant relationship. So even though at age 15 they weren't as insulin resistant, the level of free fatty acid at that age predicted what was happening later. So that kind of suggests that there might be a cumulative effect. And then not shown in these slides, if you looked at the relationship of free fatty acid levels in the 22-year-olds, it only related to their insulin sensitivity. But in the parents who are older, it also related to their blood pressure, their lipid levels, so their LDL, and some other markers of cardiovascular disease. So there's kind of a cumulative progression. So in summary, the relationship between free fatty acids and insulin resistance increases over time. And free fatty acid levels in the adolescents predict later insulin resistance. So this is the end of the experimental stuff, and now moving into some clinical application. So I'd like to talk a little bit about insulin resistance in kids, how we measure it, why we would measure it, and when. Obviously this is, hold on a second. All right, we're back. Obviously, this is something we think about in the obese population or overweight population. So first, let's talk about measuring insulin resistance. And there was a large group that did consensus examination of what kind of evidence we have and made recommendations. And Alan Snyka was part of this group. So first is discussing the different ways that you can measure insulin resistance. So I've talked a little bit in this talk about the euglycemic hyperinsulinemic clamp. We call that the gold standard, but for practical purposes, it's a research exam. It's not something that is feasible or doable in a clinical setting. The frequently sampled IV glucose tolerance test and the steady-state plasma glucose are similarly research-based measurements where you get very high fidelity, but again, very time-consuming and not feasible clinically. So the oral glucose tolerance test is something that you can do clinically, and it has a fair correlation with the clamp. Problem is, in the pediatric population, there's good data that if you look at the same child over and over, you don't have great reproducibility in the oral glucose tolerance test. So plus minus, I mean, it gives you a hint, but it's not the most reliable test. And then the next is fasting insulin. So there's looking at different studies, there's a fairly wide range of how well the fasting insulin correlates with the clamp measurement. So the R value is anywhere from 0.4 to 0.9. And with the frequently sampled IV glucose tolerance test, you also have a wide range as bad as 0.2 and up to 0.8. So it is somewhat helpful, but not great. And then finally, the HOMA. So the HOMA, actually, if you read literature, there's a lot of discussion of the HOMA as measurement of insulin resistance. And partly it's because it's a lot easier to do than the clamp or the frequently sampled IV glucose tolerance test. So for the HOMA, you basically just multiply the fasting glucose times the fasting insulin. And then there's a correction factor. But if you kind of think about what happens with someone's fasting glucose and insulin, there's a wide range in insulin that you might see in someone's fasting level. It might be from 2 to 100. Whereas with glucose, you kind of get a range of, usually in the 80s to, in somebody who's diabetic, maybe up to 120s, 140s or higher. But in your obese population, you're probably talking somewhere from 80 to 105, right? So the variation in the glucose is a lot lower than the variation in the insulin. And just mathematically, when you multiply those together, you basically get a number that reflects the insulin. So it's highly correlated with the fasting insulin, as would be mathematically predicted. So it really doesn't give you more information than the fasting insulin, but it's really popular in papers, so it's good to know about. And then people might ask, well, what about the A1C? But it's actually not a measure of insulin resistance, and we'll kind of talk about how the A1C fits into this a little bit more. So this group looked at, should we be screening for insulin resistance? So the first problem is there's no accurate, reliable, and easily applicable method of measurement, as I mentioned in the previous slide. And the second is there's no pharmacological treatment for isolated insulin resistance. Okay, so can't measure it that great, not really a clear treatment for it. And they make the really valid point that obesity itself should prompt intervention to improve weight, lifestyle measures. So whether or not you look at the insulin level in a child who's overweight, you should still be making the same advice, and that is that they should do lifestyle intervention. Okay? So their conclusion was that screening for insulin resistance alone was not warranted. So when you see this, you might think, well, I thought there was some screening that we're supposed to be doing with obese kids. And what that is that's screening for diabetes and not specifically insulin resistance. So it's important to think about when you're talking about diabetes, you're not really talking about exclusively insulin resistance. You're talking about a relationship between insulin resistance and insulin secretion. So this graph here is a really famous graph. On the Y side, well, in endocrinology anyway, on the y-axis, you have insulin secretion, and on the x, you have insulin sensitivity. And this here represents people with normal glucose tolerance. So basically, the less insulin sensitive someone is, the more insulin secretion they need to be able to achieve to maintain normal glycemia. Okay? So you might have somebody who's quite insulin sensitive and someone else who's quite insulin resistant. Neither of them have diabetes. Both of them are fine as long as their pancreas is able to be up to the task. But on the other hand, there's a spectrum that goes all the way out to diabetes where people become progressively less able to mount an islet response to the amount of insulin needed to keep their blood sugar normal. And that's where you start falling off of that curve and getting impaired glucose tolerance and then eventually diabetes. So to remember that it's a spectrum. It is insulin resistance is a key part of it, but it's not the only part. And so when you talk about screening just for insulin resistance, you're screening for something that may not actually be a problem for this person, depending on their pancreas, their islet, their genetic background, et cetera. So more to the point is really talking about the diagnosis of diabetes and when should we be screening and how do we screen. So looking at the ADA's, darn it, I don't want to join the ICE. Okay, hold on a second. All right, so looking at the ADA recommendations. So the A1C, as many of you may know, has recently been added to the criteria for diagnosis of diabetes. I put it in parentheses because that really is an adult recommendation. And while it's still part of the recommendations that applies to everybody, in the pediatric world, there's increasing realization that it might not be the best screening test for kids. And we'll talk about that a little bit more. So there's basically two ways of approaching it. There's patients who are symptomatic, okay, meaning having symptoms of hyperglycemia. And for them, you can check, you can do either a fasting plasma glucose, a oral glucose tolerance test where you define diabetes by their 2-hour plasma glucose level, or you can do a random plasma glucose, and if it's high enough, that together with the symptoms constitutes a diagnosis. But if, on the other hand, you have someone who does not have symptoms, you need to have 2 repeated values to prevent a spurious diagnosis. So you can either do fasting plasma glucose or you can do the oral glucose tolerance test. The recommendation is that you would do the same thing twice, not do one and then the other, just to kind of get one of each. But that you're going to have better fidelity if you look at the same twice. So then there's the group where maybe is more applicable to the pediatric population, and that is prediabetes, so people who are at increased risk. So this is defined by impaired fasting glucose, so that's a glucose between 100 and 125. And just to note, the WHO has a little bit different range that they use, but the ADA uses the 100 to 125. And then there's also impaired glucose tolerance. So that's a 2-hour level that's not normal, but not in the diabetic range, so 140 to 199. And then again, there's an A1C range of 5.7 to 6.4, which we'll talk about a little bit more how that relates to pediatrics. But it should be noted that in adults, there are some studies looking at that pre-diabetes range and showing that even if people do not get to the criteria of diabetes, there's still some evidence that they have some microvascular changes associated with chronic glucose abnormalities. So it's not bad enough to be diabetes, but it's not normal, and there is likely some damage going on. And so that population deserves attention and also deserves intervention, trying to move them ideally into the normal range, but at very least to keep them from progressing. So thinking about kids, it's important to talk about sort of what numbers are we talking about. So this is the estimated incidence of diabetes, and this is both type 1, type 2, and then monogenic forms. In children, this is per 1,000. And this is broken down both by age. So the lighter purple is under 10, and the darker is over 10, 10 to 19. and also by ethnicity. So it's white, African American, Hispanic, Asian Pacific Islander, and American Indian. And I think the things to note from this chart is, number one, the under 10 has a lower incidence than the 10 to 19. And that the other is that there's a little bit of difference between different ethnicities. But I think more interesting is looking at how these patients break down. So of these individuals who are being diagnosed with diabetes, what percentage of them are type 1? That's, again, the lighter purple. What percentage of them are type 2? And then this is other, so that includes the MODIs and some of the other more unusual forms. And I think one of the biggest take-home points is if you look at the under 10 population, there's very little type 2 diabetes. And of the people who have type 2 diabetes in the under 10, they are, it's much, it's barely a blip in the white population. And it's the Asian Pacific Islanders and the American Indians where you have a little, but still the majority is type 1. On the other hand, when you look at the 10 to 19 population, you see that type 2 diabetes starts to become a bigger percentage for the children of African American, Hispanic, Asian Pacific Islander, and American Indian. And American Indian is really notable in that in that age group, the majority of patients with a new diagnosis of diabetes are type 2. They're still type 1, but the majority are type 2. And so that should really color sort of how you're thinking about who you're screening and what their chances are. And that leads us to the criteria. So the first is that the age of initiation for screening for diabetes is 10 years or onset of puberty if that's earlier. And that just reflects those graphs I showed you that under 10, you're not going to really have a significant rate of type 2. Not that it's not possible, but that the rate is quite low. So then the population we're talking about are kids that are overweight. And the way you would define that can be either by BMI, by the weight per height for height percentile, or the percent over ideal weight. So any of those are considered to be valid ways to define overweight. And then not all overweight. So I think in practice we see is sort of overweight equals screening. But the recommendations are actually then to take another step and look at risk factors. So risk factors are a family history of type 2 diabetes in a first or second degree relative. And then race ethnicity, which basically breaks down to all the non-white ethnicities on their graph. So the Native Americans, African Americans, Latinos, and Asian American. So that's another risk factor. And then the third is one that we see a lot of in terms of as a consult. So kids who have signs of insulin resistance, which might be acanthosis nigricans, it might be hypertension, dyslipidemia, polycystic ovarian syndrome, or ovary syndrome, and small for gestational age by birth weight. So those kids are at an increased risk of type 2 diabetes. And then the next is a maternal history of diabetes during that child's gestation. So they get one count for having a first-degree relative with potentially type 2 diabetes if it's type 2. And then they could potentially get a second count for that being present during the pregnancy of that child. And then the frequency of screening would be every 3 years. And we kind of talked about this a little bit. That you're screening for diabetes, you're going to pick up some who might be in the pre-diabetes range, and that may mean that you would likely want to be looking at them more frequently than every 3 years. So you've got somebody who's kind of showing to be on that spectrum. So how do you screen them? So there's the fasting plasma glucose, and that's actually recommended by the ADA. There's the 2-hour oral glucose tolerance test, which is also perfectly fine. It's just more expensive, more time-consuming, requires a separate visit, although plasma glucose may as well if they're not fasting. And then there's the hemoglobin A1C. So the cutoff for children, the 6.5, that's now part of the criteria for diagnosis, it's not very sensitive. So if you don't meet that cutoff, you might be missing a lot of kids. Now, of course, if they're over 6.5, then you've got the diagnosis. That's great, or not really, but that you get them in the group. But if they are under 6.5, you don't necessarily rule them out. So, and there is some data that if their A1C is over 5.7, they have increased risk of future diagnosis of prediabetes or diabetes. But it is a little more problematic just because it's less robust in kids and less well studied for that matter. So the recommendation really would be to use the fasting plasma glucose, or potentially the oral glucose tolerance test if you have that ability. But the fasting plasma glucose is sufficient. There's no clear utility for obtaining a fasting insulin level. We do see that a lot, but is mentioned in the previous slides, it's not really clear that. So you can imagine two kids, one with fasting insulin of 10 and one with 100. And their risk of diabetes is not necessarily equivalent to those two insulin levels because it really matters how much, or those two insulin levels because it matters how much pancreatic function they have. And the person with the 10 might be closer to actually getting to diabetes than the 100. And then the A1C might be useful as a supplement, but should not be used alone to screen for type 2 diabetes. So, in conclusion, oops, let me do that. So the oxidative stress in adipocytes is associated with inflammatory changes in the macrophages. That oxidative stress in adipocytes is associated with the protein modifications, protein carbonylation, as well as serum free fatty acid levels in humans. That there's evidence that serum free fatty acid levels in adolescence can be associated with later development of insulin resistance in adulthood. And that moving into the clinical sphere, measurement of insulin resistance itself is not warranted in the pediatric patients, but screening for type 2 diabetes is recommended in selected subsets of overweight children. And finally, the A1C alone is not an adequate screening test, but it may be combined with other recommended screens, and we do use that sometimes for purposes of tracking sort of over time how the A1C might change, but for actual diagnosis, it's not adequate. And that is my presentation. I'd be happy to entertain questions.
Just take questions if you repeat the questions for the recording.
Yes, I can do that. Yes, in the back.
Presentation. Thank you.
Thanks.
I had a couple of questions for you. One, you mentioned no this medication for insulin resistance. Comment on that one.
Right.
Number two is, I go back all the way back to 2007. There was some paper published about norms for waist circumference and whether that's rising in overweight. So I left it all. It seems like that would help tease out those people who are higher than high, but who aren't overweight.
Yeah, those are both great questions. So the first question was essentially, what is the role of metformin in kids with insulin resistance? So to start with that one, so when you look at kids who are so obviously type 2 diabetes diagnosis is a separate issue, and metformin plays a major role. But in the kids who are pre-diabetic, or even kids who are not necessarily pre-diabetic but are obese, there's been some studies looking at the use of metformin in those cases. So metformin does, in one study, particularly in, I think it was Latina girls, it seemed to help with weight. It's not clear if that's partly because a lot of people get a little bit of nausea from metformin, if it's an appetite suppression. But it seems like there's more to it than that, actually. Right now, and then there's other studies looking at how do you compare metformin to lifestyle intervention in those kids who are pre-diabetic and obese. And it looks like the two really, that metformin is not clearly superior to lifestyle intervention. In fact, there's now the Today study where they have actually I don't remember what their N is, but a really large population of type 2 kids. And in that, you see that that lifestyle is helpful in addition to metformin. So I think we all kind of know that lifestyle is sort of where it's at. Metformin can also be helpful, and there's debate in the field in terms of should you be doing metformin when lifestyle intervention is risk-free or virtually, in a sense. And I think, practically speaking, it kind of comes down to the individual, how high risk you feel them to be, how likely you feel lifestyle intervention to be playing a major role. Can you try it first? And then there's some kids where you might, with obesity and a pre-diabetes picture, might say, you know what, we're just going to go straight for metformin. It depends a little bit on this picture. And then there's kids on some of the psychotropic drugs. We see a lot of actually psychiatrists starting the kids on metformin, and I don't know that the data is great for that, but that's kind of another situation where you see it. I still think in that case there's maybe other interventions that might be safer or first line. I don't know, and Brandon, would you add anything to that in terms of your feeling on metformin?
Not really. I think you summed it up pretty well. I think the data on improvements in markers of insulin resistance are really mixed in this population when they're placed on metformin. So I think it's kept people a little bit lukewarm at using it sort of universally
And I think there's also, I have a little bit of a bias of, that's one of the few things that are approved for type 2 in the pediatric population. And so I feel a little bit like I don't necessarily always want to start with my biggest gun. And then the second question, remind me again, it was about waist circumference. So that's a really good question. So there are people who've done some work looking at, can you take the overweight population and further tease out information that might be useful in figuring out people at higher risk? And we certainly know that waist circumference or central adiposity is higher risk than just any generalized adiposity. When you talk about the concept of the metabolic syndrome, which is starting to fall out of favor, waist circumference is part of that. I would say, generally speaking, that yes, somebody who has a relatively bigger waist circumference is going to be at higher risk, but there isn't at this point any sort of well-studied, this is the criteria at which point you are more likely to put them here or there. And it doesn't fall into the criteria for screening for type 2. And actually, I'm not sure that when they made those criteria, if there was enough information to even really consider waist circumference in that algorithm. So, but I think it's an interesting point, and in my mind, that's part of the gestalt of like who is the higher risk individuals versus the lower risk individuals. But great question. Any other questions? Yes, Harvey. Yeah, so that's a great question. At what point you should be screening. And a fair amount of time, people, when they're doing the sort of the overweight workup, will do lipids, looking at fasting blood sugar, but also looking at liver enzymes to kind of get a feeling for that. One of the problems with NAFLD, and you'd be able to speak this better than I, is that there's not great diagnosis measures. I mean, you can do a liver biopsy on everybody that you suspect, but that would probably be a little overkill. So how to tease it out, and there's certainly probably kids who have the beginnings of NAFLD who might not have liver enzymes abnormalities too. So what ends up happening, I think, is as part of that general looking for complications of obesity, I think it makes sense to do the liver enzymes at very least. And then at that point, kind of depending on, and I think if a child was obese enough, you might even say, step it up and do a liver ultrasound. But I think that typically the recommendation would be just looking at the liver enzymes and then sort of going from there. But it's definitely a great question. I think an active part of research is looking at how to get our hands on that question and how to better pick out the kids that might be at risk. Yes. Yeah. So as I just repeat that the liver enzymes are pretty easy to do. So. Yeah. Yes.
So we're getting a beautiful presentation first off. I was curious, struck by some of the slides, the one study that you did between high-fat mice that were fed high-fat diets and the OB mouse, and how leptin signaling might also play a role in this pathogenesis between carbonylation proteins, and does impaired leptin signaling increase the amounts of that, or do those levels actually impair leptin signaling, since obesity is a state of leptin resistance?
Yeah.
How does that actually play a role, and how does that fit into that whole pathogenesis?
Okay, so the question is, based on the basic science looking at the OB mouse, the leptin knockout, if there's any information or thought about how leptin might be playing a role in carbonylation and that aspect of pathogenesis. And the quickest answer is there's no information to my knowledge. So carbonylation in adipocytes thus far has only been, the only publications I've seen are from the Bernler lab itself. So, and they have not looked at leptin signaling, so I don't think that anybody's looked at that. In this case, the knockout mouse was kind of used as a, so the OB mice don't have to be on the high-fat diet to get obese. So it kind of teases out the overweight from the being on a high-fat diet that causes you to be overweight. So it gives you just two different models of obesity. But it is an interesting question. Sort of leptin is is a very tantalizing molecule because it had, everybody kind of got excited that all we have to do is get leptin and everybody can be thin and it'll all be solved. And then unfortunately that really only held true for mice and not for humans where we have leptin resistance. Does carbonylation relate to leptin resistance? Again, it's not been studied, but I think that's actually a really sort of the diverse effects of oxidative stress. You certainly could hypothesize that might be one of the ways. And I think what really is going to happen is that there's not one pathway that connects oxidative stress to the metabolic changes. There's multiple different things that happen, and sort of oxidative stress is a common trigger or common sort of help signal from the fat cell. So, any other questions? All right. Well, thanks so much for your attention. I really appreciate it. Exit. Did I speak like 50 miles an hour? Oh. And loudly?